Hello students, welcome to the biology class. So today in the last class we learned about the process of oogenesis. We had some amount of differentiation or we learned about the differences between the oogenesis and spermatogenesis. Then we learned about the hormonal control over the process of oogenesis. So let me quickly revise these topics that we can move over to the next topic. We learned that something common in both the gametogenesis. Gametogenesis is the process of formation of gametes. So we learned there are two types of gametogenesis. In the female body it is oogenesis, and in the male body it is spermatogenesis. We learned that the common thing is that all of these processes involve MGM, multiplication, growth and maturation. Multiplication involves mitosis, growth involves feeding, and maturation involves meiosis first and second, followed by a certain amount of cell differentiation. The first difference which we learned was that during the oogenesis starts early in the fetal stage, somewhere around 25th week of the pregnancy, the oogenesis starts, but spermatogenesis starts at puberty. Second difference we learned was that oogenesis involves two arrestations. One arrestation occurs at prophase first or diakinesis, and the second arrestation takes place at metaphase second. But in the case of spermatogenesis there are no arrestations. Third difference we learned was that during oogenesis the primary oocyte, when undergoes division, it develops into two cells which are unequal in size. One is called secondary oocyte, this first, and the other one is called the primary or the first polar body. And when the secondary oocyte also divides, it divides unequally into oocyte and the second polar body. So the process of maturation or meiosis first as well as second both are with equal karyokinesis but followed by unequal cytokinesis. So unequal cytoplasmic division is there. But in the case of spermatogenesis, we learned in those classes that the primary oocyte divides by meiosis first to form secondary spermatocytes which are equal in sizes, and the secondary spermatocytes divide again by meiosis, that is meiosis second, to form spermatids, and they are again equal in size. So there is equal cytoplasmic division in meiosis first as well as second. The next difference we learned was that spermatogenesis starts in the testes and finish in the testes, that means inside the seminiferous tubule, but oogenesis starts in the ovary and it gets over in the fallopian tube. So there are many more differences. If you wish you can learn, like for example during oogenesis primary oocyte will give rise to only one egg, but in the case of spermatogenesis primary spermatocyte divides to give rise to 4 sperms. Next difference, oogenesis starts early, so it's a process which starts early also, and the formation of a mature egg requires years because it starts in the fetal stage and it has to wait till puberty, so it requires years to be finished. But the process of spermatogenesis, we learned in the last classes, is finished in 64 days. Generally one egg is released every month, and in this case there are millions of sperms which are released. So there are many differences. One more you can learn, that oogenesis involves stoppage. It is going to finish at somewhere around the age of 45 to 50, and that is called as menopause, and there is no such thing like menopause. In the case of the spermatogenesis, it continues till the point of the death, even in an old or aging male human. So we learned some differences. Now let's move on that what exactly happens during the follicular development, because follicle is something which is confusing. So we learned that during the oogenesis topic, that when a female is still developing in her mother's body during the embryonic development, there are egg nests developing which differentiate into primary follicles. Now primary follicle carries one primary oocyte inside it, surrounded by the follicular sheath. Remember, these follicular sheath cells are supposed to release estrogen later on. So these primary follicles, they carry the primary oocyte which was about to finish its meiosis but could not because it is arrested at diakinesis. Now when she enters puberty, these primary follicle will undergo further changes. So the follicular sheath will change into granulosa cells, and theca develops because of the cortex outside the granulosa cells. Then the secondary follicle changes into tertiary follicle with the development of a fluid-filled cavity called as antrum, and theca differentiating into theca externa and theca interna. Theca externa is fibrous in nature and theca interna is cellular. Now at this stage, please be careful regarding it. At this stage,